In Rudyard Kipling's story, How the Leopard Got Its Spots, we learn that the spotless leopard asked his friend to paint him with spots so that he could disguise himself among the rocks and leafy branches and better sneak up on his prey. This is one of a series of similar just so stories that Kipling crafted. And it's relevant to this discussion because biologists are often accused of telling just so stories when we devise beneficial or adaptive explanations for pretty much everything, like the giraffe's long neck, the desert fox's huge ears, or the spines of the prickly pear cactus. But this adaptationist perspective, while compelling, in which most features are seen as conferring benefits, really can't tell us everything. We know that natural populations harbor large amounts of genetic variation, and understanding how this variation enters and leaves the population is critical to understanding how populations will adapt to environmental change. Some would argue that we can't really understand genetics if we see all features from an adaptationist perspective. Rather, we need to realize that sometimes who lives and who dies is due to random or stochastic events. Mike Lynch developed predictive models to help us understand this process in different contexts and for different species. So let's talk about Mike Lynch. Mike was born in Auburn, New York. His parents didn't go to college, but he went to nearby St. Bonaventure for perfectly understandable reasons that they had the #1 basketball team in the country. At St. Bonaventure, he developed an interest in freshwater ecology, so it's no huge surprise that he ended up at the University of Minnesota for his PhD. It also was apparently a well-informed decision. He studied with Joe Shapiro, and some of Mike's work with plankton stimulated a growing interest in genetics. But he took a faculty position in limnology, really, at the University of Illinois, where he notes that making the transition from hardcore limnology and into population genetics bordered on insanity in terms of getting tenure. Well, he did that. He may have been insane, but he got tenure. And then, at his next position at the University of Oregon in Eugene, Mike continued his genetics work. While he was there, he started thinking much more about the role of deleterious or harmful mutations. He was also involved in debates about how to protect the wild salmon population. It was in Eugene that he started forming his ideas about mutational meltdown. So what is that? Here it is. I'll give you a minute. Basically, deleterious or harmful mutations can sometimes occur faster than breeding can eliminate them, and the overall fitness of a population decreases. This is likely to lead to a decline in population size, which leads to further accumulation of deleterious mutations, and the cycle continues, spiraling into what Mike called mutational meltdown. He says once you cross a certain threshold of population size, there becomes the risk of extinction. Organisms today must cope with a wide range of anthropogenic disturbances, from habitat loss and pollution to nutrient deposition and climate change. If natural populations are to adapt to these rapid environmental changes, what should we be shooting for in terms of population sizes? Probably somewhere around 5,000 to 10,000 breeding adults for an adaptively secure population. But many of our endangered species aren't even listed as such until they're well below this number, suggesting that interventions are needed much earlier. An example would be this Kirtland's warbler, whose worldwide population dipped to just a few hundred individuals in the 1970s. And what about captive breeding and release of these individuals into wild populations? A potential problem here is that captive environments are very different from those in the wild. These young Galapagos tortoises are well-fed, protected, and taken care of when they're sick. The genetic variants that would help them survive in the wild are not necessarily the same as those that are favored in captive conditions. Such programs may be raising individuals that are genetically less fit when they're released into the wild. This suggests that for the long-term genetic health of wild populations, captive breeding programs should only be used as a last resort. Clearly, increasing effective population size is key. Providing lots of habitat and connecting existing habitat may be the best option. For example, little over 1% of native prairie remains. In the last few years, prairie conservation efforts in Minnesota have shifted to focus on increasing not just the amount of preserved land, but connections between these existing prairie remnants. In this way, 77% of Minnesota's remaining native prairie would be coupled with linear stretches of restored habitat that would function as 6-mile-wide corridors to connect the individual fragments. In addition to these plans to protect prairie remnants, effective population sizes are critical for conservation of the Powashik skipperling, which used to be common here but is now down to fewer than 500 individuals. Also, the peregrine falcon, often hailed as a conservation success story, and the winged maple leaf mussel, currently being reintroduced into local stretches of the Mississippi River after having been extirpated. In these ways, Mike's work on mutational meltdown has come home to Minnesota, and tonight, so has Mike. So hopefully you will also stand and let us acknowledge you.